Welcome to the first video in our new series on understanding science. These videos are designed to help you be able to determine whether or not a research article contains good science or bad science. In today's video, we're going to be talking about controls. So when we talk about controls in scientific experiments, especially when we're talking about papers relating to multiple sclerosis, there's three types of controls that you'll mainly see. We're going to try and explain this using fruit. I know you've heard that you can't compare apples and oranges before, but we're going to try and use that today to explain how this works. So three types of controls that you might see. One, you might see where we're comparing different types of MS. So for example, you may see a paper where they're looking at relapsing or remitting MS versus secondary progressive MS to try and see the differences between the two. This is like comparing a green apple with a red apple, for example. In this case, we're looking at MS, so we can't really infer anything about MS as a disease as a whole, but we can certainly infer what might make the green apple different from the red apple. So for example, we might want to see whether or not green apples are sweeter than red apples. We can only do that by comparing these two apples. On the other hand, you may see experiments where they compare multiple sclerosis with other neurological disorders. So for example, here we have a green apple with a pear. They're both the same colour, they're roughly the same shape, but there's obviously differences between the two. This helps us really try and cancel out all of the side effects that may be happening because there is a disease process going on. So instead of just looking at things that may be caused by the inflammation or the fact that there's cell death going on or other factors like this just looks at factors that may be specific to what's causing the MS. So this is quite a good control to have. The other control that you may see more often is this example of comparing apples and oranges, where you have people with MS versus healthy controls. Now this obviously has some benefits, but also some negatives, just like I referred to when I talked about why we use other neurological disorders. So obviously this looks then at what's the difference between someone who has MS versus someone that's healthy, what's actually going on in the disease. The difficulty in using something like this is that it does pose that problem of, is this something that's actually causing MS, or is it just something that's happened because someone has MS? It's the chicken and the egg effect, and that can often be something that's quite difficult when looking at this sort of control. So while that may tell us whether or not the controls are valid in terms of the sort of experiments that you're looking at and the outcomes that the research has generated, you also need to look at how the controls have been picked. So if we go back to that first example, say, of looking at the differences between two different types of MS, or the red and the green apple. It's really important that you're comparing apples, in this case, that are as close together as they can be, so that really the only difference that you're looking at is the fact that one is red and one is green. So if we look at these apples here, these are quite well matched. However, if for example we wanted to do an experiment, say, where we were trying to work out whether red apples or green apples were more filling, if we compared these two, we might be able to make a conclusion out of that. But if, for example, instead we were looking at these two, still a red and a green apple, but clearly the red apple is far smaller than the green apple, we really couldn't make any judgments out of this because there's too many variables in there. Obviously, the green apple is probably going to be more filling just because it's bigger, not because it's a green apple versus a red apple. It's the same when we look at controls, and you may see when you read papers or see articles about paper that they talk about age-sex matched controls. And that's really trying to get the two groups that you're comparing against as close together as possible, so that you're taking out factors such as gender, such as age, such as levels of disability, such as how long a person's been dealing with MS. So not only do you need controls, but you need them to be as closely matched as possible so that the outcomes that you generate are as valid as they can be.